Hi, good day everyone. You're welcome to my YouTube channel, Everything Medical. Today's video, we shall be looking at the glycolysis, part B glycolysis. I've done video on glycolysis, so that one is part A. So today's class is part B of glycolysis. We have talked so many events that took place when glucose is being oxidized in the cell cytosol, that is glycolysis. Before I dive into this class, if you are coming across my channel at the first time and endeavor to subscribe so that you get notification when I upload video on medical courses. Now let's proceed. We have done video on part A, which is what I said previously. We have given a definition on the glycolysis, the cellular compartment of glycolysis, the organ of metabolism of glycolysis, the significance of glycolysis. We also gave a concept on the difference between the isoenzymes of glycolysis, which is the hexokinase and cokinase. We also went further to define or gave the regulations of glycolysis, both its hormonal regulation, gene regulation, and allosteric regulation. Now, we shall be delineating on the biochemical reactions that do take place on glucose oxidation, right? So now let's proceed. First of all, before we proceed, let's give a recap on the concept of glycolysis. Glycolysis simply means breakdown of glucose. As you can see here, this is glyco, which is glucose. This is lysis, which is breakdown. So it simply means the breakdown of glucose to produce pyruvate, lactate, and ATP. That is the simple definition of glycolysis. So it is a catabolic pathway, that is breakdown. It is a catabolic pathway by which glucose is being broken down to produce what? Lactate, pyruvate, and ATP. You have to understand that this pathway occurs in the cytosolic part of a cell. So it is a cytosolic pathway, which means that all the enzymes are cytosolic enzymes. You can see my diagram here. In here is the cell cytoplasm, while outside this place is the extracellular part of the cell. That means the environment that is not inside the cell. That is the cellular compartment of the glycolysis. It occurs in the cell cytosol. So since it occurs in the cell cytosol, its enzymes are called cytosolic enzymes. Now, likewise, the organ of metabolism of glycolysis. We said previously on my part A video that glycolysis occurs in every organ. It occurs in every organ, right? Because every tissue requires ATP for cellular respiration, for energy breakdown, right? Every tissue requires energy for their daily function. So glycolysis is the only metabolic pathway that occurs in every tissue in the human body, because every tissue requires ATP, which is energy for their cellular respiration, maintaining their daily task, right? Now, this is the pathway that depicts the biochemical reactions of glycolysis, right? Now, before we delve into these biochemical reactions of glycolysis, glycolysis can be called EM pathway, right? EM pathway, EM pathway. It's simply the scientists, those scientists that founded it. So they were named after the pathway. So EM means Enden-Meyerhof pathway. So it can be called Enden-Meyerhof pathway. Likewise, glycolysis can also be called the Pannas pathway. Pannas pathway. It can also be called the glycolytic pathway. All these things are just for glucose breakdown. It is just for glucose breakdown. There's nothing more. to produce ATP, which is energy, because glucose is the most abundant carbohydrate molecule we have in the human body. So that is why it features in giving tissue energy for cellular respiration. Now, when you take carbohydrate contained food, right, it gets digested in the GI tract. Once you get digested, it will be absorbed, particularly in the small intestine, which is the ileum. The ileum is the largest part of the small intestine. That is where absorption takes place. Once it is being absorbed in the small intestine, particularly the ileum, now at the intestinal lumen, those nutrients, which is the glucose, enters the bloodstream from the intestinal lumen. Once it enters the bloodstream, there will be a signal to the pancreas to secrete insulin, because insulin has to be secreted to regulate the blood glucose level. Already, those glucose have not entered into any cell, right? They have not entered into any cell for metabolism. Now, once insulin is secreted, insulin will open the glucose channels in every cell, right? And also activate some certain glucose transporters which are under the influence of insulin. It is not all glucose transporters that are under the influence of insulin. It's only GLUT4 that is under the influence of insulin. That is why in a diabetic patient, GLUT4 is usually deranged, because in a diabetic patient, insulin is not available. That is why GLUT4 activity is usually deranged. So it is those glucose molecules that are not insulin dependent are usually at work, such as from GLUT1, GLUT2, GLUT3, GLUT5, and GLUT7, right? So now this cell here, this is a cell. This is the cell cytosol, because this pathway, which is glycolysis, occurs in the cell cytoplasm, right? So this is the cell membrane. This place is the extracellular part of the cell, that is environment that is outside from the cell. In here is the cell cytoplasm, right? Now, when those glucose has been found in the blood, they will begin to be transported by a specific glucose transporters into different organs, because every organ have their own specific glucose transporters, right? Now, here is the cytoplasm of a liver cell, which is the hepatocyte, right? So we'll be making reference to the hepatocyte, which is the liver cell. Now, this is glucose here. This is glucose, which is found on the extracellular part of the cell. This is glucose. And glucose is a hexose sugar, right? It has 6 carbon atoms. Look at it here. 1, 2, 3, 4, 5, 6, right? Now, once glucose is found at the extracellular part of the cell, it requires a transporter, which will transport it from the extracellular environment to the intracellular environment, right? And that transporter is the transporter that transports glucose to the liver, because we are making reference to the liver cell, right? And the transporter is GLUT2 transporter. This GLUT2 transporter is not under the influence of insulin, and it transports glucose to the liver cell. Transport glucose to the liver cell. So this GLUT2 transporter will transport glucose from the extracellular environment of a cell to the intracellular environment of the cell, which is the cell cytoplasm. This is glucose here. Glucose has been transported. Now, before I proceed, what is glucose transporter? These glucose transporter are membrane proteins. They are called membrane proteins, which facilitate the transport of glucose from the extracellular environment to the intracellular environment, right? They are membrane proteins because they are found on the cell membrane. So the membrane proteins will transport or facilitate the transport of glucose from the extracellular environment to the intracellular environment, right? Now, glucose has been transported in the cytoplasm of the liver because we're making reference to the liver hepatocyte. Now, once glucose has been transported, what will happen next? Glucose will be phosphorylated into glucose 6-phosphate by an enzyme called hexokinase or the glucokinase. Glucose will be phosphorylated to glucose 6-phosphate. Glucose 6-phosphate by an enzyme called hexokinase or glucokinase. In this step, which is the step 1 of glycolysis, right? 1 ATP is utilized to phosphorylate glucose to form glucose 6-phosphate. So ATP is utilized. ATP will be utilized to phosphorylate glucose, right? So it is 1 ATP that is utilized to phosphorylate glucose to form glucose 6-phosphate. Recall that glucose have 6 carbon atoms, right? What will happen on that phosphorylation is that phosphate group will be attached on the carbon number 6 of the glucose structure, forming glucose 6-phosphate by utilizing 1 molecule of ATP. Now, these enzymes uses magnesium ion as a cofactor. Magnesium ion as a cofactor, right? Now, these enzymes are isoenzymes of glycolysis. They are isoenzymes of glycolysis, right? Now, they have their distinguishing features. They are distinguishing features of hexokinase and cokinase. This cokinase is found in the liver only. That is why we call it hepatic glucokinase, right? Why hexokinase is found in every tissues in the body. So that means it is found lesser quantity in the liver, whereas this glucokinase is found mainly in the liver. That is why we refer it as hepatic glucokinase. So it is liver. This one is found in every tissue, right? Or tissues. This hexokinase phosphorylate all hexo sugars. It phosphorylates all hexo sugars. Why the glucokinase only phosphorylate glucose? So glucose is phosphorylated in the liver to form glucose 6-phosphate by the utilization of 1 ATP, because ATP is reduced to form what? ADP, right? You have to understand that this glucokinase still have its 6 carbon atom. They still maintain their 6 carbon atom. 1, 2, 3, 4, 5, 6. Same thing with glucose. The only change is that there is addition of phosphate group to the carbon number 6 of glucose. That is why it changes to become glucose 6-phosphate. Now, this step is the irreversible step of glycolysis, although it is being circumvented by the key enzymes of gluconeogenesis, which is called glucose 6-phosphatase. That is the key enzyme of gluconeogenesis, which circumvent the key enzymes of glycolysis, which circumvent the key step of glycolysis, which is step 1. Step 1 is the key step of glycolysis because, as you can see, it is not reversible. It is irreversible step of glycolysis that is being circumvented by the key enzyme of gluconeogenesis, which is called glucose 6-phosphatase enzyme, right? Recall that this glucose 6-phosphate have its own fate because once it is being generated, it has other metabolic fate, right? Where it features in other metabolic pathway. What are the fate of glucose 6-phosphate? We have the fate of glucose 6-phosphate. We have HMP shunt. HMP shunt, we have glycogen metabolism, glycogen metabolism, we then have glycolysis itself. Yes, glycolysis itself. So these are the metabolic fate of glucose phosphate, right? Now, this glucose phosphate will be isomerized to form fructose phosphate, right? To be isomerized to form what? Glucose phosphate. That means the reaction that undergoes in step 2 is isomerization reaction, while step 1 is what? Phosphorylation reaction. And the enzyme is phosphohexose isomerase, which generates fructose phosphate. Fructose phosphate will be generated by an enzyme called phosphohexose isomerase. It is this enzyme that will isomerize glucose phosphate to form fructose phosphate. They still maintain that 6 carbon atom. You can see 1, 2, 3, 4, 5, 6. They are still hexose sugar. Now, fructose phosphate will be phosphorylated to form fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate will be formed by the phosphorylation of fructose phosphate by the enzyme called phosphofructokinase enzyme. And in this step, 1 ATP is utilized to phosphorylate fructose phosphate to form fructose 1,6-bisphosphate. So ATP is utilized to form ADP. 1 molecule of ATP is utilized. The enzyme is called phosphofructokinase. PFK. This step is an irreversible step of glycolysis. It's an irreversible step of glycolysis. Recall that step 2 is a reversible step. Step 3 is an irreversible step of glycolysis, right? And this enzyme called phosphofructokinase is an allosteric enzyme of glycolysis. It's an allosteric enzyme of glycolysis. Now, step 3 is an irreversible step, which is being catalyzed by an allosteric enzyme. PFK is an allosteric enzyme. It's also an inducible enzyme and also a regulatory enzyme of glycolysis, right? And this PFK it has an activation and also inhibitions. That means it has an activator and inhibitors, right? What are the activators of PFK? We have the insulin, we have the AMP, we have fructose phosphate, we also have fructose 2,6-bisphosphate. Those are the activators of phosphofructokinase. We also have their inhibitors, those molecules that inhibit its activity, such as the glucagon, the cyclic AMP, the low pH, the citrate, and ATP. Those are its inhibitors, right? Now, this step 3 is an essential step of what? Of glycolysis. The enzyme, or what is the key enzyme of glycogenesis which circumvents this step? It is fructose 1,6-bisphosphatase. It is the key enzyme of glycogenesis which circumvents this step, the step 3 of glycolysis. So that is why gluconeogenesis is the opposite of glycolysis, but not a reversal of glycolysis, right? Because the key enzymes of that pathway, which is gluconeogenesis, do circumvent the key steps of glycolysis, right? Now, we have generated our fructose 1,6-bisphosphate. What will happen next? Fructose 1,6-bisphosphate will be cleaved by a lyase enzyme to form 3 carbon molecules compound, right? Which is G3P and DHAP, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These are the 2 molecules that will be generated as a result of the cleavage of fructose 1,6-bisphosphate by an enzyme called aldolase enzyme. It is a lyase enzyme, right? So the enzyme is aldolase enzyme. We cleave this molecule to form 3 carbon molecules because fructose 1,6-bisphosphate is a 6-carbon molecule, right? Once it is being cleaved, it will form what? 3 carbon molecules, which is 1 and 2. So it will generate what we call glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate will be generated, and DHAP, dihydroxyacetone phosphate. Dihydroxyacetone phosphate, acetone phosphate. That is DHAP generated, dihydroxyacetone phosphate, G3P, right? So once fructose 1,6-bisphosphate has been cleaved by an enzyme called aldolase, it is a lyase enzyme to generate G3P and DHAP. DHAP we channel to the liver to form glycerol. Now what? DHAP will be dehydrogenated to form glycerol 3-phosphate. This is it here. Glycerol 3-phosphate, glycerol 3-phosphate by an enzyme called glycerol 3-phosphate dehydrogenase. So that reaction is a dehydrogenation reaction, right? That means NADH is generated. NADH plus H will be generated by an enzyme called glycerol 3-phosphate dehydrogenase. Glycerol 3-phosphate dehydrogenase enzyme. We dehydrogenate DHAP to form glycerol 3-phosphate. Now, this glycerol 3-phosphate will be phosphorylated to form glycerol. So this is glycerol. by an enzyme called glycerol kinase. That means since it's a phosphorylation reaction, ATP is what is generated, right? Now, glycerol will be stored in the liver. During glycolysis, this glycerol, which is a neutral fat, will be mobilized during glycolysis to form glucose. That is why one of the significance of glycolysis says that this glycolysis provides carbon skeleton, which is used for the synthesis of amino acid and glycerol part of fat. Look at it here. Glycerol is a neutral fat, which is being produced by glycolysis pathway. So once it is being produced from DHAB, it will be stored in the liver. Now, during glycogenesis, it will be mobilized back to produce what? Glucose. So that is the concept of it that led to the formation of glycerol in the liver. As you can see, this arrow, it is forward and backward. That means they are all reversible, right? They are all reversible. And the reversible aspect of it is what? Gluconeogenesis. Because during that occurrence of the pathway called gluconeogenesis, glycerol will be what? Mobilized back from the liver to produce what? Glucose for tissue respiration. Now what? DHAB will be reconverted back to follow the forward reaction by joining what? Glycerol. By joining glycerol 3 phosphate by an enzyme called phosphotrioseisomerase. That is the enzyme that will convert DHAB to follow the forward reaction. So we have phosphotrioseisomerase, isomerase, which will convert DHAB to follow the forward reaction, right? Now, glycerol 3 phosphate will be dehydrogenated and simultaneously phosphorylated to form 1,3-bisphosphoglycerate. Look at it here. To form 1,3-bisphosphoglycerate. To form 1,3-bisphosphoglycerate, right? So, since it's a dehydration reaction, NADH is produced because it is a what? A dehydrogenation reaction, and it undergoes a simultaneous reaction by phosphorylation. That means there is presence of inorganic phosphate, which is an energy bond for the forward reaction. So NADH plus H is generated, NADH plus H generated, and NAD plus is reduced because this is the reduced form of, this is the oxidized form of NAD plus. Why this is the reduced form? This is the one that enters the ETC for the production of energy, which is electron transport chain to generate ATP. And 1 NADH equals to 2.5 ATP, right? 1 NADH that is being generated in this step equals to 2.5 ATP. 1 NADH equals to 2.5 ATP in new calculation and 3 ATP in old calculation, right? So The enzyme that catalyzes this step is called glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. That is the enzyme that catalyzes this step, right? And this step, which is step 5, it is the bottleneck of glycolysis. We call it the bottleneck of glycolysis. Step 5. Bottleneck of glycolysis. Is the bottleneck of glycolysis. Now, step 6. 1,3-bisphoglycerate will be phosphorylated to form 3-phosphoglycerate. Look at it here. They have 3 carbon atoms on their molecule to form 3-phosphoglycerate by an enzyme called bisphosphoglycerate kinase enzyme. You have to understand that step 6 of glycolysis is called the substrate level phosphorylation because energy is trapped from a molecule without referring to the complicated electron transport chain reaction, right? Because 1,3-bisphosphoglycerate will be trapped to produce a molecule of ATP, a molecule of energy, which is called what? ATP, right? Without referring to the electron transport chain. So, the enzyme here is bisphosphoglycerate kinase, bisphosphoglycerate kinase. So energy is being produced. That means ATP is being produced. ATP will be reduced to produce what? ATP. This step is the substrate level phosphorylation of glycolysis. The substrate level phosphorylation because ATP is generated without referring to the electron transport chain. Now watch. Step 1 and step 3 we utilize our ATP. No energy is generated. Whereas in step 6 we produce energy, which is ATP. You can see my arrow, right? See my arrow on this, on how it is being fashioned, because on this step 1, ATP was utilized to produce ATP. Same thing applicable with this. That is why step 1, 2, and 3 are the preparatory phase of glycolysis. Whereas step 4 and step 4A are the splitting phase of glycolysis because we split our molecule, which is fructose 1,6-bisphosphate, right? Now, step 5 to step 10 are the energy within phase of glycolysis because in this phase, we generate energy, we generate ATP. We don't utilize ATP. But here, we utilize ATP. We lose our energy. But in this phase, we generate our energy, right? That is why this phase is called the energy within phase of glycolysis. Now, we have said earlier that step 6 is the what? The substrate level phosphorylation of glycolysis because energy is being trapped from a molecule without referring to electron transport chain. Now, in respect to oxidative phosphorylation, right? In that area, there is a production of ATP in electron transport chain from the reduced equivalent, such as the NADH, which is here, NADH plus H. Because this NADH plus H do enter the ETC to generate ATP. That is why we call it oxidative phosphorylation, because ATP will be produced from the reduced equivalent, which is this NADH plus H in the electron transport chain. Whereas in this case, ATP is produced without referring to the electron transport chain, right? Now, 3-phosphoglycerate will be isomerized to produce 2-phosphoglycerate. Look at it here. All of them are having three carbon atom on their molecule to produce 2-phosphoglycerate by an enzyme called phosphoglycerate mutase. So it is a mutase enzyme. Mutase enzyme, right? You have to understand that this bisphosphoglycerate kinase, which is the enzyme that catalyzes step 6, uses magnesium ion as a cofactor. So there's a magnesium ion here as a cofactor. Now, 2-phosphoglycerate will be reduced to form phosphoenol pyruvate. Phosphoenol pyruvate to form phosphoenol pyruvate. Now watch. This is step 8 of glycolysis, right? This step 8 is being catalyzed by an enzyme called enolase. Enolase enzyme. Enolase enzyme is a magnesium ion dependent enzyme. It uses magnesium ion as a cofactor. So it is a magnesium ion dependent enzyme. And one molecule of water is liberated or removed. One molecule of water is removed. Right? Now, this step 8 has a clinical significance because while determining the blood sugar level, right, the physician usually administer fluoride. How does the fluoride inhibit the activities of enolase? Now what? Fluoride, when you administer fluoride before checking the blood glucose level or the blood sugar level, that fluoride will go to step 8 of glycolysis and back off magnesium ion from binding with enolase. Once it does that, that fluoride will irreversibly inhibit what? Enolase, thereby making the glycolysis pathway to become stable. Then you can check or determine the blood sugar level. If you do not administer fluoride, this pathway will keep up running because it will continue to run, thereby breaking glucose down. So the main reason to administer the fluoride is to make the glycolysis pathway become stable. Then you can check the blood glucose level, the blood sugar level. That fluoride will back off magnesium ion from binding with enolase because this magnesium ion is a cofactor to what? To enolase, right? So once it back it off, it will do what? It will irreversibly inhibit enolase enzyme. So that is it. Now, phosphoenol pyruvate will be dephosphorylated to produce pyruvate. This is pyruvate to produce what? Pyruvate. To produce pyruvate, right? By an enzyme called pyruvate kinase. All of them are having three carbon molecule. You have to understand that ATP is generated here. So it is also a substrate level phosphorylation of glycolysis, right? ADP to ATP. And this enzyme uses magnesium ion as a cofactor and even potassium. Magnesium ion as a cofactor. And also potassium ion. This step is an irreversible step of glycolysis. Step 1, step 3, and step 9 are the irreversible step of glycolysis, right? Now, step 3 is circumvented by the key enzyme of glycogenesis, which include pyruvate carboxylase and phosphoenol pyruvate carboxykinase, I mean to say, right? There are two enzymes of glycogenesis which circumvent the step of glycolysis, which is this step. Those enzymes include pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So those are the key enzymes of glycogenesis which circumvent the key step of glycolysis, which is particularly the step 9, right? Now, pyruvate will be reduced to lactate. This is lactate. by an enzyme called lactate dehydrogenase. And this reaction is a dehydrogenation reaction because NADH is generated, which enters the electron transport chain to produce ATP. So we have the oxidized NAD to reduce NAD by lactate dehydrogenase, which is LDH, lactate dehydrogenase enzyme. Right? Now watch. We have two types of glycolysis, aerobic glycolysis and what? Anaerobic glycolysis. From glucose to pyruvate is aerobic glycolysis. So once pyruvate is generated during aerobic glycolysis, pyruvate will be channeled to citric acid cycle for complete oxidation. Now, during particularly anaerobic glycolysis, right? Lactate will be channeled to Cure cycle, right? Lactate will be channeled to what? To Cure cycle during anaerobic glycolysis. Lactate will be channeled to Cure cycle because the Cure cycle is the cycle that relieves the muscle during strenuous exercise, because during strenuous exercise there is usually an accumulation of what? Lactic acid in the muscle, which leads to muscle cramp or fatigue. So during that process, there is usually an occurrence of Cure cycle, which relieves the accumulation of lactate or lactic acid in the muscle, right? So that the muscle can generate glucose for its contraction. Now, during that aerobic glycolysis, pyruvate will be channeled to what? To TCA cycle or citric acid cycle for complete oxidation of glucose. That is why the ATP that is generated during glucose complete oxidation is 32 ATP and or 38 ATP in old calculation, because 32 ATP is for new calculation and 38 ATP is for old calculation. New calculation, old calculation. This is the number of ATP that is used for complete oxidation of glucose, which involve glycolysis and citric acid cycle. Because for glucose to become completely oxidized, there is usually involvement of glycolysis and TCA cycle, because pyruvate has to enter the TCA cycle for its complete oxidation, right? Now, you have to understand again that 7 ATPs are generated during aerobic glycolysis, while 2 ATP are generated during what? Anaerobic glycolysis. Those are MCQ question in medical board exam. So we have done justice to the metabolic pathways of the glycolysis, right? I believe you enjoyed the class. So here comes the end of this class. We have done justice to it, ranging from how glucose enters the cell cytoplasm. It got broken down to generate glucose phosphate. It has its own metabolic fate. We form our pyruvate, we form our lactate, right? Okay. Don't hesitate to subscribe and also share to your friends in medical college. Thank you so much for staying to the end of the class.